today's Research Connect seminar series. Thank you and welcome. My name is Sean Delaney. I am a CDTRP member and a longtime patient partner. I'm sure many of you have seen me before. I'm a transplant professional working with the Alberta Transplant Institute, as well as a two-time kidney transplant recipient and lifelong renal patient. I'm grateful to the CDTRP to be part of this webinar. First off, I'd like to acknowledge that both the CDTRP and my own Alberta Transplant Institute are both hosted by the University of Alberta, which is located on the lands of the Treaty 6 territory. This is the traditional and ancestral territory of the Cree, the Dene, the Blackfoot, the Saulteaux, the Nakota Sioux. I also acknowledge that this territory is home to regions 2, 3, and 4 of the Métis Nation of Alberta within the historical Northwest Métis homeland. As a backcountry hiker, skier, and camper in the eastern slopes of Canada's Rocky Mountains, I'm regularly reminded of the many ways that my history is intertwined with the history of the original inhabitants of this land. I acknowledge the many First Nations, Métis, and Inuit who have lived on and cared for these lands since time immemorial. I'm grateful for the knowledge keepers and elders who are still with us today and those who have gone before us. I make this acknowledgement as an act of reconciliation and gratitude for every Indigenous family on whose territory I am blessed to live on, work on, raise a family on, and recreate on. I encourage you to write your own land acknowledgements in the chat box if you would like to. We'd also like to acknowledge that Takeda, AstraZeneca, and CIHR are generous sponsors of this seminar series, and without whose support we could not host them. Before we dive into the main content, there's a few CDTRP updates. Just a reminder that our annual scientific meeting for CDTRP is less than 2 months away. It will be in a hybrid format with the in-person component in Montreal on December 5th through 7th. The registration is now open and folks can register on the CDTRP website. A link will be added to the chat box shortly. And just finally, before I introduce our fantastic speaker today, a few housekeeping notes. Today's session will be recorded and posted on the CDTRP website as well as their YouTube channel. During the presentation, please keep your mics off. If you have questions or comments, you can raise your hand with the raise hand function or type your question into the comment section of the Zoom window. I'm pleased to introduce our presenter, Dr. Caroline Lamarche. We just had this conversation. Dr. Lamarche is a clinician scientist and transplant nephrologist at Maisonneuve-Rosemont Hospital. She is an assistant clinical professor at the Université de Montréal. After her nephrology training at the University de Montréal in 2015, she completed a master's in using adoptive immunotherapy to treat and prevent PK nephropathy in kidney transplant recipients. She then pursued a postdoctoral fellowship with Dr. Megan Levings at the University of British Columbia on using chimeric antigen receptor regulatory T cells to induce transplant tolerance. Her lab is working on developing adoptive immunotherapy in nephrology and understanding Treg dysfunction. We are all very excited to hear you speak. Over to you, Dr. Lamarche.
Thank you, Sean. I'll share my screen. Here, just switch 1 minute. Okay, is that good?
We're seeing your speaker notes actually.
Oh.
Yes, perfect. That's the one.
Okay, perfect. So hi everyone, thanks for having me today. So I'll talk to you about Treg exhaustion, myth or reality. And it seemed really easy question to answer, but it's not that easy. So I was asked to present you today the results of our latest paper. So it's a paper that was published finally in last May, and is a result of the work I've done under Megan Levings during my postdoctoral fellowship. So I have to say I have conflicts of interest because I'm co-inventor on patents on A2 CAR Tregs, but I don't think it's too relevant for this talk. So today the talk will be about Tregs, and if you can have like one key message from this talk is that Tregs are the good guys in transplant, and we love them. I love them, and I hope you'll love them after this talk. So Tregs are really so they're very good to mediate transplant tolerance, and they're good to control other immune cells that could be overactive and mediate rejection. And that can be in solid organ transplant, but also seen as graft versus host disease after bone marrow transplant. So our goal is to do Treg adoptive immunotherapy. And why we want to use cells instead of pills to mediate tolerance is because right now we have to admit we don't have good pills. So the drug that we use to stop and prevent rejection right now in transplant, they come with a lot of side effects. So they can increase cardiovascular diseases through hypertension, diabetes, and cholesterol. And they can also decrease the immune system and lead to infection and cancer. So we think that by using cells, it would be more physiological and we won't have all of those complications. And by using Tregs, we would be able, so our goal is to be able to give less immunosuppressive drug. We know that we won't be able to stop all of them, but at least if we can decrease the side effect and the amount of drugs. So what we want to do is to collect Tregs from a solid organ transplant patient, or it can be from a bone marrow recipient. I think it would be more from their bone marrow donor in that case. But it can also be from third parties. We know at the CDTRP that Megan Levings and Laurie West are working on Tregs from thymuses, from kids. So that could be a possibility here. So we want to collect Treg, but Tregs are very rare and we won't have enough cells to treat a patient just by collecting some and reinfusing them. So we'll need to expand them in vitro. And we might also want to modify them. So I'll talk a bit more about that later. And then we'll reinfuse them. How do Treg work? So this slide is very busy and it's not meant for you to read everything, but the real answer is that Tregs can mediate tolerance through many different mechanisms. And we don't really know which one is the most important. And it's probably like this mechanism is important under one circumstance and this other mechanism in another time. So one of the things that Tregs can do is metabolic disruption. So they have this CD25 here. So it's a high affinity IL-2 receptor, and they're really good to grab all of the IL-2 in the media. So then the other conventional T cell will starve without IL-2. So they can also do cytolysis, so directly destroy other cells. They can secrete inhibitory mediators. That's quite important. So for example, IL-10, TGF-beta, and adenosine here, because they have those receptors to transform ATP, CD39 and 73. One of their main mechanism of action is through their action on dendritic cells, and they will lead to a more immunotolerant DC phenotype. And this is a way to continue tolerance. They can also lead to induce Tregs, so convert conventional T cell to more to Tregs, induce Tregs. So we know now from many mouse studies that Tregs are important for tolerance. And that's just one example, one of many. But we know in mouse model of transplant that if we remove Tregs, we can't develop tolerance. So that was a heart transplant model, but it's been done with many other models. We also know that injecting more Tregs can induce graft tolerance. And once again, this is only one model, but I think in animals, it's been well proven. I haven't shown here, and it's important for people that are working on bone marrow transplant, but injecting Tregs can also decrease graft versus host disease. And it's a very well known model to test the efficacy of our Tregs. Now we've moved past preclinical studies, and there are already many phase 1 and 2 clinical trials using Treg adoptive immunotherapy that have been published and are on the way. My question here was, are all Tregs the same? And how can we compare the quality of the cells between different trials? So for those of you who are clinicians, we're really used to compare in the study when you compare drugs to compare the protocol. So what's the dose of the drug? What's the timing? Was it one dose, multiple dose? What was the control group? Were there even a control group? What were the immunosuppressive drugs, the amount, the tapering? What's the endpoint? Is it a biopsy, rejection, number of immunosuppressive drugs? All of those things are important to judge the quality of the study. However, now when you use cell as a therapy, this adds a whole level of complexity. So for example, for Tregs, the source is important. As I said, Laurie and Megan are working on harvesting Tregs from thymuses. So thymic Tregs. You can use Tregs from peripheral blood and even cord blood, but they might not be the same. So it's something you need to consider. Also, who is the donor? So if you use autologous cells, for example, in transplant, it means that you harvest cells from a very sick patient, either before transplant, while he's under dialysis, for example, or after transplant while he's on immunosuppressive drugs. And all those things will have an impact. If you use allogenic or third party, cells will be healthier to start with, but then because they're allogenic, they might not survive as long because you'll have an allogenic reaction and your immune system will want to attack these cells. What's the lineage? So there are two types of Tregs, natural and peripherally induced. Natural one comes from the thymus, and they're more stable. So they're demethylated in the promoter of FOXP3, TSDR region. And we know they'll be more stable. If you use peripherally induced, then they might convert back to conventional T cells. Are they naive or not? Usually when you use naive Treg, they might be more pure and more natural. So I won't explain it all here, but it's something that needs to be considered and stated in the study. How did you isolate your Treg? Was it a magnetic separation? Were they sorted? What's the percent purity? And you'll be surprised in the next slide where it's not always very good. Then what's the specificity? Are they polyclonal or antigen specific? We know that if you use polyclonal Treg, about up to 10% of the cells should react against the graft. And it's something that's been seen in some study where after injection, you see only a few clones that survive and then grow. So some of them are not useful. If you use antigen-specific Treg against the graft, maybe you'll need less Treg to have the same impact because they can all react to the graft. Then were they genetically engineered? Do they express on CARs or transgenic TCR? And what were the release criteria and the extension protocol? So what was considered okay to infuse to a patient? Was it 80% FOXP3 positive, so 80% like Tregs, or 90 or 50 or even lower? What was the mortality, the viability of the cells, all of those things. So this is also a very busy slide and it's not meant for you to read everything. It's just one example. So those are all published studies, all phase 1 and 2 clinical trials that use Treg adoptive immunotherapy and solid organ transplant. Here, if you have a look here, those are the 2 oldest studies. And the cells, if you really look at the study, the cells that were actually injected to patients in this one, only 15% of them were really Tregs. And we don't really know what were the other cells. So how can you judge of the outcome if you don't really know what you injected? This one is even worse. So it was 3.6% that were really Tregs, and the other one we don't really know. So we saw an impact that was not that good. So half of patients had rejection. What does it mean? I don't know. Then you have studies here that use no induction therapy. So the one study is like a consortium of multinational trials that compare cell therapy and transplant. And in those studies, they replaced standard induction therapy. So either chemo, it was basiliximab. So instead of injecting basiliximab to patient, they gave some cells. But in some other study, they injected thymoglobulin, which can be really toxic for Tregs before infusion. So you also need to consider that. And were cells injected before transplant, 5 days after, 2 months after, or even in the TASK trial, they didn't inject Treg to prevent rejection. They tried to use them to treat rejection. So it was in patients with active rejection. So all of this is highly variable. And here in this talk, I want to add one more layer of complexity, as it's not complex enough. So I'm sorry for that. So I want to talk about T cell dysfunction. There are many types of T cell dysfunction. One of them is senescence. So what is immunosenescence? It's when the cells are aging, becoming older. So they usually have shorter telomere. They have lower proliferative potential because they have cycle cell arrest. They usually have also they accumulate DNA damage. Yeah, and so there's not a lot of study about senescence in Tregs, but it seems that Tregs can become senescent and maybe at a faster rate than conventional T cells. So there's even a group that suggests that with aging, because Tregs seem to age faster than other conventional cells, that's why in the old people, you'll have higher, for example, Th17 ratio, less Treg, and you'll have a bit more inflammation because of that. So with the inflammaging, so more inflammation as you age. What are the triggers for senescence? It's mostly age, but also like stress and chronic inflammation. And it's something to consider in if you take autologous Treg, so Tregs from patients that have been really sick with a lot of inflammation in their life. So they might not be as good as other Tregs. Then there's another type of dysfunction that's called anergy. So usually cells become anergic when they have TCR stimulation, but without signal 2, so without co-stimulatory stimulation or cytokines, and then they don't proliferate as well. This is not really studied in Tregs because many people consider Tregs to be anergic compared to conventional cells. So that's a bit of a debate just in that. So today I'll focus on exhaustion. So exhaustion comes from a persistent antigen exposure, same as human. So if you're always active and we always ask you to work, at one point you'll get exhausted. Then what you'll do is that you'll express those inhibitory receptors. You won't be able to proliferate as much, and you'll secrete less cytokines. Maybe more prone to apoptosis. So what is known about exhaustion? So it's mainly defined in CD8 T cells, but I think now it's well accepted that it also exists in conventional cells. So it's really because of a persistent antigen stimulation, and that can be really antigen exposure or CAR signaling. So it's something I'll come back after because that's a model we use. Exhausted cells have reduced production of cytokines, don't grow as much, and express inhibitory receptors. And they do that because they have a transcriptional reprogramming, an epigenetic reprogramming, and a metabolic reprogramming. So what have we learned from studies that's been done in cancer? Because we know that immunotherapy has been used a lot in cancer, more than in transplant. So we have to study what they've done and their issue in order to improve our product. So we know that if you take conventional T cells that are exhausted and you inject them to patients, they won't persist as long and they won't be as good to fight cancer. So what about Treg? And Tregs become exhausted. So it's not that easy to answer because Tregs to start with are really similar to exhausted Treg at a starting point. So Tregs do express those inhibitory receptors, and they use that to mediate their tolerance. So for example, they express CTLA4 that can bind to CD8086 on the surface of a DC. And that's one way to make DCs become more pro-tolerance. So those receptors are one way that they are good Tregs. Tregs do not secrete IL-2 and interferon gamma. So you can't use the reduced cytokine expression to say that they're exhausted Tregs. And they do have a reduced proliferative potential compared to conventional T cell. Now, what was known before we did that, so there's a few studies that suggest that exhaustion do exist in Tregs. For example, in this study, so as you probably know, we now have a lot of checkpoint inhibitors, so a lot of drug that can target exhaustion. So if you target exhausted conventional T cell, they'll become more like better conventional T cell and they'll fight cancer. So anti-PD-1 therapy is one of those. And they realized in gastric cancer patients treated with anti-PD-1 therapy that instead of clearing their cancer and getting better, 10% of patients had hyperproliferative disease, so became worse after anti-PD-1 therapy. So they studied those and compared them with patients that responded to therapy. And they saw that they had a lot of PD-1 positive Tregs in their cancer. And after anti-PD-1 therapy, their Tregs became hyperproliferative. So also like better cells, but because they had better Tregs was not as good for cancer. There's also this study that suggests really something similar, although I don't trust this one because they, without showing any gate, they said that in some patients they had like 40% Tregs. And I'm a bit skeptical because usually it's like Tregs are like 5% of CD4. So 40 seem a lot. So I don't know if they really gated on Tregs. So in order to study exhaustion in Tregs, we use a tonic-sealing CAR model. So we work in collaboration with Crystal Nickel. And so they showed in this model in multiple really good papers, so Science, PNAS, Nature, Nature Med. So they have this CAR with tonic signaling, that's the HA CAR. So this CAR at the surface of the cell is always on. So it's the same as persistent antigen stimulation, but very, very strong. And they showed that this CAR can lead to exhaustion, and their control is is the typical CD19 CAR that's been used in clinic to in cancer. So they showed that their tonic-sealing CAR lead to spontaneous phosphorylation of CD3 zeta, that their cells with the tonic-sealing CAR express a lot of the typical markers of exhaustion, PD-1, TIM3, LAG3, secreted less cytokine, had epigenetic reprogramming, and had the same cytotoxicity in vitro, but decrease efficacy in vivo. So we sorted naive Tregs, stimulate them, transduce them to express the CAR. At day 7, sorted Tregs that did express the CAR, and keep them in culture until day 11. So we had 3 group untransduced, so without any CAR, the CD19 CAR, who does not have tunneling and should not be exhausted, and the HA CAR, or tunneling 1. Then we studied the impact of this on Treg phenotype, transcriptome, so RNA, chromatin accessibility, methylome, metabolism, and function. What we saw is that there were a lot of similarities between exhausted Tregs and exhausted TCON, but also some differences. So our Tregs expressing the tunneling CAR here in green did express lots of LAG3, TIM3, and PD1, but also a lot of GITR and 4-1BB, and more CTLA4, LAC, and GARP, who are more like Treg markers that are important for their suppressive effect. This was without destabilizing Tregs, so they continued to express FOXP3. FOXP3 is Treg main transcription factor. And they were still demethylated in their TSDR, so those were natural Tregs and they stayed Tregs. We sorted naive Tregs in order to have more natural, and they still express Helios. Then we studied their transcriptome, and we compare Tregs versus TCONV and Tregs between themselves. So when we compare Tregs versus TCONV, the good news was that Tregs were still Tregs. So it was great that they were stable and they were still doing their purpose. So less interferon gamma, granzyme, and more FOXP3, Helios, and GARP, really typical of Tregs. What was interesting is that they had a different shell expression of inhibitory receptors. So compared to conventional cells, the exhausted one, the one with penicillin, had lower PD1 and LAG3, but more TIGIT and CTLA4. So they do express inhibitory receptors, but maybe not the same as conventional cells. And they also had a unique transcription factor signature. So more tox, which is one of the main driver of exhaustion, BAF, PRDM1, which codes for BLINT1, and less AOMACE and TBET. Now, if we remove the differences between Tregs and TCONV at baseline to only study what was different in the one that expressed tunneling CARs, we also saw less of some gene and more of some gene. And when we studied Tregs between themselves, we saw some interesting things. So more of some transcription factors such as NR4A1, and less of, other one, but that's okay. Then when we looked at hallmark pathway analysis, we saw a lot of things that came out was about metabolism, and that was very interesting. So way more like MYC, mTOR signaling, glycolysis, oxphos, cholesterol, fatty acid, adipogenesis. So it was mostly about metabolism. So then we decided to do seahorse assay to study a little bit more their metabolism. So this assay, it's not perfect, but it gives us an idea of what's going on. So you can use ECAR, so the extracellular acidification rate, as a surrogate for glycolysis, and the oxygen consumption rate as a surrogate for oxphos. And what we saw is that Tregs with tunneling CAR started way over in both. So they're like more energetic metabolically active. But the favored ratio was between on glycolysis, so more glycolytic. So they started more. Here, I think I have it, yeah, in the other side. When you normalize them at baseline, you see here that they start higher, but when you push them with the drugs, it can't increase. So they really have a limited spare glycolytic capacity. So they're all in metabolically, but you can't push them furthermore. And that was interesting because I didn't say, but our Treg, tonic-linked CAR Tregs were more apoptotic. So maybe they're so metabolically active that at one point they'll go into apoptosis. Then we studied their ataxia, and we saw a global increase in chromatin accessibility. Some genes that are of interest for us, such as BLIMP1, were more accessible. No changes for toxin in our 4A. That was a bit surprising because it's something that came out in our RNA-seq. And some of the changes were Treg specific. When we looked at the transcription factor motif and we compared our signature to what was found in conventional T cell by Crystal Mickle's group, it was really, really similar. So enrichment for P1, NF-kappa B, RONX, and FAT family members. So this makes sense, but I didn't know when I started that. But I thought that chromatin accessibility was quite stable, but I realized it really isn't. So it's something that can change in a matter of hours. So here it's just one study, but I like this one. If you look chromatin accessibility here in pink, it can change in a matter of hours. It really goes well with RNA here, but DNA methylation is something that's way more stable. So in order to really define the signature, we decided to study DNA methylation. For this, we collaborated with Crystal Mickle at St. Jude. And they developed a multipotency index. So they studied DNA methylation of exhausted CD8, human CD8. To do that, they use CD8 that are specific for HIV. So it's really a persistent, like a natural model of persistent antigen stimulation. And they developed that index to say, so those are really exhausted, those are naive, and that's the spectrum. What we realize is that Treg to start with are more exhausted on that scale compared to T-con. But when we compare our two groups of Tregs, they were really similar. So for DNA methylation, in 12 days, we didn't see much change, to be honest. Tregs are a bit enriched in those genes that can define exhaustion at baseline, but it didn't change. So Tregs more exhausted than T-con, and that's mainly what we saw. We saw some genes that were more methylated. So when it's more methylated, it's very less transcribed. So in TCF7, so that's like naive, and less methylated in TOX and PRDM1, which was interesting for us because they're quite typical of exhaustion. And we also saw them at a protein level. Now, so a lot of data, but now how does it translate in their efficacy? So first we tested them in a suppression assay. So that's typical how we test Tregs. So we test them in a 4-day suppression assay in vitro. We look at their ability to suppress the proliferation of CD4 and CD8 Tregs. And in fact, very surprisingly, our tonic-linked CAR Tregs were more suppressive. So at least as suppressive or even a bit more suppressive than other Tregs. So they express all of those inhibitory receptors, but because those receptors are important to mediate their action, they're effective. However, when you look at the Treg division index, so if they divide through the 4-day culture, they divided less. So they don't grow as much, the ones that are exhausted. Then we decided to inject them into mice to look at graft-versus-host disease. So we took mice that are immunocompromised, we irradiate them, we inject them with human PBMCs, with or without Tregs. So here you can see that all the mice that only receive PBS survive. All the ones that receive only PBMC, so without Tregs, they died. Almost all of them, we stop here. The one that receive untransduced Treg, they all survive. Tregs are really good to prevent graft-versus-host disease, but the one that receive our CAR Tregs died. So our Tregs were not suppressive in vivo. So the conclusion of this study is that I think that Tregs are susceptible to exhaustion, but the signature is Treg specific. So I won't go through all the details, but I think that's the thing you want to remember. So they are susceptible to exhaustion, but it's Treg specific. Now, as for the next step, it would be nice to have an easy exhaustion signature in Tregs. I don't think we can only take PD-1 positive to define them as exhausted. I think what's important here, if we want to move towards Treg adoptive immunotherapy in transplant, is to study at least write it down in the study. It's exhaustion signature, and if it's important for its activity in patient, maybe add this as a release criteria for the different studies. And I want to work more on finding a way to reverse and prevent exhaustion. So I want to thank Megan Levings, because this project was done in her lab, and all the people that helped me, Crystal Michael, Caitlin Zebley, Michael Cover's lab, German for the bio info, and all of our partners. And here are my lab members from my lab that's now continuing this project. And I hope one day I'll have the data from my independent lab to show you.
That was fantastic. Thank you so much, Dr. Lamarche. I'll open the floor to any questions. Just a reminder, you can use the raise hand function in Zoom or post your questions into the chat for discussion. I see a few of my fellow patient partners on the line. Hans, Donna, Ellen, great to see you as well. But I'll start off with the first question with a patient hat on. So with the CDTRP and other agencies all having a shared vision of one transplant for life, I can see how your work is, bringing our world and our patients' care one step closer. I guess I think all of us as patient partners would love to see a future with lower immunosuppression requirements or eliminated. Of course, that's an ideal. But if you were to project out, how far out can you see the work that you're presenting on today and the future work of your lab moving from kind of animal and early human trials to standard for care?
Yeah, so I have to say that right now there are a lot of studies being done all around the world using Treg adoptive immunotherapy, and we don't have it in Canada for now. So as I see it, is that the first step would be to participate to some study in Canada with even like the baseline product, because I think we need to go quickly and not miss the boat. So we need to go with the current version of the cell product, and then the work of my lab is to work also to develop the next phases. But in Canada, we're not even in the boat right now. So we need to start there. And one day, if we have this ongoing here, maybe I can work to improve the cell product and then move on to different steps. So I didn't talk today about the other work that I've done with Megan. So we worked to develop A2 CAR Tregs, so CAR Tregs that are specific for the transplant. And at least I have to say that our construct is now being tested in clinical trials in Europe. So it's not in Canada, but at least we developed something that's being tested in patients right now, just not here.
Yeah, no, I mean just the baseline knowledge between your lab and Lori and Megan and everybody is, I mean, fantastic to see. But yeah, we can definitely have a full foot in this field going forward. That would be fantastic to see. I see a question from Carolina.
Hi, thanks so much for that great talk. I was just wondering, what do you think is the physiological or kind of natural role of exhaustion in immunology? If we think about like T conventional cells, CD4s and CD8s, it makes sense that they get exhausted as a way of controlling excessive inflammation. But with Tregs, what do you think is the physiological role of exhaustion?
It's a very good question. So yeah, for T conventional, as you said, it's quite clear because at one point, if you don't get exhausted, you have too much of a response. For Tregs, I don't know if it's a way to control the other way around. So if, for example, because yeah, to stop too much, like can it be a way to, if you don't have that and your Tregs are too powerful against one antigen, can they lead to cancer or persistent infection? I have no idea. I have no idea, but it's something that would be interesting to study for sure.
Thanks so much for for attempting to answer.
I don't know. So it's the thing as well. Like so we proved it in an artificial model, so using CARs. So we studied it in an artificial model, and I think that's relevant for cell therapy because that's an artificial model. But right now I have no clear sign that it does exist in the natural patient, except those two studies with checkpoint inhibitors that their cancer had a flare up. So I don't know if it's something that can happen in a natural way in vivo. If it makes sense.
Thank you.
There's always the default answer of more study required. Do we have any other questions for Dr. Lamarche? Please share your camera or put your question in the chat.
So I want to apologize if it was a bit too basic. I didn't do it more much for patient partners, or I went right to the paper, but yeah, don't feel free to reach out with even basic question. There is no bad question. No bad question.
Absolutely. We've not seen any questions appearing. Oh, there we are. Miss Erica.
I was trying to get my camera to work. Sorry about that. I have a basic question. Let's go for it. CD25, you're saying it's a receptor of the T cells. Is it specific to T cells, or they comprise other cells of the body?
So they're more on Tregs than other T cell. I think it's only on T cell, but I have to check. So it's more on Tregs than T conventional. So that's why, and it's a high affinity. So that's why they catch IL-2. It's upregulated in activated T-conf. We can Wikipedia it. As I said, there's no stupid question. I don't see. But it makes sense that it's mostly T cell because it's an IL-2 receptor. So.
And then you also mentioned early on one of your
On
slides.
B cell, NK, it can be expressed as well on activated cells.
Yeah.
Yeah.
You mentioned also earlier you had a murine study. Is Tregs different in humans and mice?
Yes, they are. And that's something that we really need to be careful when we compare the studies. There are a few differences with natural Tregs, induced Tregs as well. Like many differences. I'm trying to think about one specific. But it doesn't act the same. So I'm really, because I'm a clinician, and I work mostly with human, I try to work more with human cells. And if some things have been proven in mice with mouse Treg, I try to want to prove it in human, and it's not always true.
Thank you very much.
Fantastic. Last call for any final questions for Dr. Lamarche. We look forward to people interacting also through the YouTube channel as well once the recording is posted. If there are no other questions, I'd just like to say thank you so much to everybody for your questions and comments throughout, and especially to Dr. Lamarche for presenting. If anyone has additional questions or suggested topics and speakers for future seminars, please get a hold of Erica at ekcroft@cdtrp.ca. And thank you to everyone, and enjoy the rest of your day. Thank you so much, Dr. Lamarche.